Hello everyone, welcome back. So we have been completing so many things in this chapter. Still so many things are remaining. Anyways, we'll be doing it as fast as possible. And yeah, today what we will be doing is very important part of this chapter. However, very important organelles we have completed already. But today what I'm going to do actually, that is still more important. Why? Because the next thing what we will be studying in this chapter, everything depends on the class what today we are going to do. And that is, we are going to study about today, it is cytoskeleton. Cytoskeleton. Yes, cytoskeleton. The name itself, I think you people will understand, right? Cyto means what? Cyto means cyto, this term. Cyto means what? Cyto means cell. And you know what is a skeleton system, right? Now we being human beings, okay, we also have this skeleton system. Okay, and every organism, okay, even if you consider it as a single cell also, even it needs that skeleton system. Now why it needs, you can just relate it with ourselves only. See, now if you imagine ourselves, like you imagine the human beings without skeleton system, what would happen? Would be able to walk, okay, we people would not have even strength. We have bones in our body, that why, that is why we have particular shape. Every organism also, if you see, even if a bird, even if an animal, if you see, they have some particular skeleton system. That is why they have that particular shape. They move because they have that strength, and that strength comes from that skeleton system. That skeleton system helps in what? It helps in maintaining the shape. It gives the mechanical strength. It helps in moving from one place to another. So same function even this skeleton does, and this skeleton which is present within the cell is called cytoskeleton. So what is cytoskeleton? The skeleton system which is present within the cell, is called cytoskeleton. Now if you, and we are talking about, particularly if we are talking, you should not get confused, we are talking about eukaryotic cytoskeleton. Now the question arises, then what about prokaryotic cell? Then prokaryotic cell will not have cytoskeleton. They don't want to move, or they don't move. Yeah, even they have cytoskeleton. But today what we are going to mainly focus is about eukaryotic cytoskeleton. Prokaryotes also have cytoskeleton, but still research is going on, study is going on. What are the proteins it is made up of, all those things still is going on. But we have a clear picture of cytoskeleton of eukaryotic cell, so it has been put into the syllabus. And we need to understand this first. So cytoskeleton which is present in eukaryotic cells, we are going to talk about that. Now one more good thing is that animal cell and plant cell. So in eukaryotic cell we have animal cell and plant cell. Both of these cells have same cytoskeleton, so there is no need to study different. Okay, same cytoskeleton we need to study. So first we will see in a crude thing like cytoskeleton functions. So according to NCERT, you remember 3 M's you have to remember to understand what? To understand the functions. What are the functions? The functions of cytoskeleton. The first function, remember 3 M's here. 3M, if you remember, then it will be easy for you. First is maintains. What it does? It maintains the shape of the cell. Shape of the cell. This is very much important. So it helps in maintaining the shape. Second M is, it helps in, what is that? Maintaining the shape of the cell. Second thing is mechanical strength. Sorry, I forgot. Mechanical strength. It provides what? It provides mechanical strength to the cell. Then what? Third one is motility. What is motility? Motility means ability to move from one place to another. It helps in movement. See, these 3 important functions you need to remember. Main 3 important functions are these 3. First is, it maintains the shape of the cell. It gives mechanical strength, and it helps in motility also. How it helps in motility? We are going to study everything in detail now. However, if you come to this function, maintain the shape of the cell. See, for plant cell, you can say that it has some particular shape. Because it has cell wall. Okay, you have studied about cell wall, you people know very well about cell wall. Now the question thing is that plant cell have cell wall, so easily plant cell will not get ruptured. Easily it will not get ruptured. That is why it is so, it has a lot of strength. What about animal cell? Animal cell don't have cell wall, but they have the cytoskeleton which functions like cell wall. I mean, which functions in giving what? The shape for the cell. Now for example, if you consider a cell, let us consider this is a cell. This is a cell. Now there is nucleus in between. Okay, and then we have this endoplasmic reticulum like this. We have Golgi bodies like this, all these things. Now these cytoskeleton, where you will find them? If you will find them completely dispersed everywhere in the cell like this. Like this. Now it forms, what you can see here, they are everywhere in the cell like this, like a long filaments. Like this, they are like filaments. So what it is doing, because it is, covering the whole cell, it is, remember this cytoskeleton is a proteinaceous structure. It is made up of some particular proteins. Again, there are types of cytoskeleton that we will see now later. But overall, if you talk about cytoskeleton, their function is what? They gives the shape. Because they are connected from nucleus till this plasma membrane. Okay, they are, they are holding the cell properly. They are holding the cell organelles properly in particular area. Also, you can say along with this, you can also remember one more thing, that is anchorage. It anchors. It encourages what? Encourage the cell organelles. That means what? It helps in, proper placement of these organelles. Now this Golgi body is present here. It will not come and just every time it will not come and dash, or it will not come and attack this nucleus every time. Why? Because cytoskeleton is present here. So it will not allow that movement. Then you will think then there are so many things which needs to move inside the cell also. Then if cytoskeleton is not allowing movement within the cell, then how? There is one more thing, that cytoskeleton also, remember, it is dynamic in function. Dynamic, for example, we have already done regarding this vesicles, no? This endoplasmic reticulum, they have ribosomes, so they produce proteins, they produce what, ultimately vesicles are formed, which needs to go to Golgi bodies for packaging, and Golgi body will pack them, and then it will send out of the cell sometimes, or within the cell, those vesicles need to move. During that time, wherever the it needs to change, that time it will allow that movement of those particles within the cell. If movement is not required, then they will become very stiff. They gives particular shape for the cell, they maintains the shape, they gives mechanical strength, all those things. That is why I said it is dynamic. Whenever it is required that it should be little bit, it should give some movement, it will give. If movement is not required, it will not give. Okay, so these three are the main things. I think you understood this first one, and also you understood this mechanical strength. Whenever some compression is happening to the cell, because cytoskeleton is present within the cell, it will not allow easy rupture of the cell. Now one more easy example I'll give you. Now whenever we do some exercises, we stretch our body, our skin cells, every time they are stretched. Whenever we do yoga, whenever we do exercises and all those cells, they are getting stretched. And if cytoskeleton were not there, for example, then the cell would rupture, no? Because of that wear and tear process. But it doesn't happen. Why? Because of cytoskeleton. And what the cytoskeleton do? They are like flexible, you can say. They are flexible. They move. They help in contraction. They help in relaxation. Both. And they give stiffness also. Likewise, they function. So, they give what? They give mechanical strength. Also, they maintain the shape. Now, how they help in motility? How they help in movement? That we will see later. We will just see this later. Now, anchorage. I think you understood here. They help in placement of organelles in particular area. Because they are present everywhere within the cell. So, particular area, those things are there. Now, I was talking about vesicles, right? So, whenever it is moving. Now, for example, if a vesicle is found here, you can see, for example, there is a vesicle over here and it needs to move through this it can easily come like this. It will help in within cell, the movement can happen of this vesicle. Just like, you know, the track. No need to get within the cytoplasm. Just move on the cytoskeleton. Easily you will get a way. So nothing is going to be complex when it is moving on the cytoskeleton. So it makes a way for that. Cytoskeleton will make a way. It acts like a track also for those material which are needed to be moved within the cell. And then dynamic, I said, that means it can change. It is always, you know, it is always in favor with cell. However cell requires, likewise it acts, or it works, it functions like that. So I hope you understood these functions, why cytoskeleton is important. Now particularly cytoskeleton is what? It is nothing but skeleton system within the cell. Then what it is made up of? It is made up of proteins. It is made up of proteins. Which proteins? Which different types of proteins are there now? Everything we are going to study, and that we will study in types. So till then, just take down this much. Okay, fine. So I'll make it clear. We are talking about eukaryotic cytoskeleton, and it is made up of what? Cytoskeleton are skeleton-like structures. Like they are made up of what? They are made up of proteins. Okay, and we saw 3 M's, 3 important functions: maintaining the shape of the cell, mechanical strength, it provides mechanical strength, and the third thing is motility, it provides movement. And we are going to still talk about that movement, how it helps in movement. Fine. So which are the 3 types of? Now let us see types of cytoskeleton. There are 3 main types of cytoskeleton in both plant as well as animal cell. You will find these 3. So those 3 are, first one is microtubule. First one is microtubule. Next comes microfilament. First one is microtubule. Second one is microfilament. And the third one is intermediate filament. We have to talk in detail about these 3 things. That is very much important. But out of these 3, what is more important for your syllabus is first one. Because if you know this in detail, you will understand the next structures, cilia and flagella structure. Okay, please concentrate. Okay, here, so microtubule, microfilament and intermediate filament, these are the 3 types of what? They are 3 types of cytoskeleton structures. Now, all these 3 are different. Why they are different? Because their function is almost same. We have seen no functions. All those functions are same. Where they function, sometimes that differs. And the main difference of these 3 is that the diameter, size. It depends on size. First thing is size. And second thing is what protein they are made up of. So that we will see here, and then we will see in detail about everything. So first of all, the first thing about all these 3. Remember, microtubule is made up of protein. And that protein is tubulin protein. Which is the protein? It is made up of tubulin protein. Then coming to microfilament, it is made up of actin protein. Which protein? Filament is made up of actin protein. Coming to intermediate, see, intermediate filaments, it is made up of various proteins. So we cannot go on naming every protein. So in total, we can say it is made up of a family of keratin protein. the same keratin which we have in our hair. Okay, same protein, keratin protein, which actually makes our hair. So we have that family of keratin protein wherein different proteins are there within that. So many proteins are there. So we can just remember it as family of keratin protein. Here, tubulin, actin, keratin. What are these three? These three are what? These three are proteins. Again, you people should not get confused, right? These three are what? They are proteins. So this is the first difference. I hope you understood how these three are different. Then comes, I told you, size. The diameter, these three, they differ in their diameter. So here, this is the largest one in case of size, if you see. And I'll just tell here, you just take down here, again I'll be telling you in detail. This is 25 nanometer in width. We will see that how it is. And then it is, microfilament is about, you can say 7 to 8 nanometer, approx 7 to 8. And this is about 10 nanometer. So how will you remember? You need to remember these two things only. Mostly, like question will be coming from these two. What is the protein they are made up of and what is their size, width. Okay. So here it is 7 to 8 nanometer. It is 10. So if you see which is the largest one, almost because it has 25 nanometer width, it will be largest. And this is smallest. And this is intermediate. So intermediate is in middle. It's not too big, it's not too small. So it is like this. So please remember these things. However, we'll be doing everything in detail so that time you'll understand. So this is about the types of what? Eukaryotic cytoskeleton. 3 types are there. 3 proteins are there. They're made up of 3 different proteins and their width, or you can say size. This thing you have to remember. Take down this much. Okay, so yes, we will see in detail now. So first one we will see microtubule, which is very important. Okay. Which are the 3 types of cytoskeleton? Eukaryotic cytoskeleton. The first one is microtubule, and then comes microfilament, and then intermediate filaments. Okay, now coming to the first one, that is microtubules. Microtubules. Okay. Now microtubules, I told you 2 important things about microtubule, which is its protein. Its protein is what? Tubulin protein. How you will remember this? Tubules. Tubulin. Easily you can remember. And what is the diameter? 25 nanometer. Okay. Now we need to study about microtubules. These microtubules, they are tube-like structures. What you can understand here? They are tube-like structures. For example, if you take any tube, any hollow tube, it looks like this. It looks like this tube. It looks like this tube. Like this. You can say it looks like a hollow tube. Okay. Now this microtubules, if you see the structure of microtubules, how it is made up of. Okay. These things, microtubules, are made up of 2, monomers. 2 monomers. Now, I'll just show you how it is made up of. It is made up of 2 monomers. And those monomers are, for example, let us consider, okay, this is one monomer and this is one monomer. Okay, like this. Now this is called, both are what? Tubulin proteins only. Monomers of that. This is called alpha. This is called alpha tubulin protein. And what about this? This is called beta tubulin protein. I am talking about structure of microtubule. Okay. Now this alpha tubulin protein and beta, what they do is they go on making a sheet-like structure. They go on making a sheet-like structure like this. together. They both join together and then they make what? They start joining the other like this. Like this. They go on joining like this. Are you able to understand what I'm talking about? Okay. Here. Like this. They go on making a sheet. Okay. Now again, next will be, they will completely form one thing, one sheet type structure. Again, next is again this. What is this? Again, this is alpha. Joining to this alpha is beta. Alpha protein, beta protein. Again what? Again alpha. And then again beta. I hope you are getting this, right? Like this, it forms a complete sheet. You know how many it forms? It forms about 13. Like this, you will find like this till 13. Like this, it goes on like this. It forms 13 alpha and beta proteins, 13. The first one what we are seeing, till 13, it forms a sheet-like structure. After that, what will happen? That sheet which is formed, it will coil. I'll just show you with the help of this paper so that you can understand easily. Okay, now let us consider this sheet. This sheet is what? Microtubule. How it is made up of? It is made up of alpha, beta. Alpha, beta. Like this, it has completed one sheet-like structure now. After completing this sheet-like structure, what will happen to this is, it will just, it will just coil like this. Like this. Are you able to understand? No, what has happened here? It has just coiled. Like this. Properly. Yeah, like this. Okay, now this is proper shape. Can you see this? It's hollow from here. You can see it is hollow, but it is made up of what? It is made up of alpha and beta tubulin proteins, which are alpha and beta tubulin monomers. Ultimately, it forms structure like this. It folds. This sheet, it will fold and form like this structure. And if you see from here, how many proteins you will see? I told you, no? How many proteins? After folding, this type of structure it has formed, right? Right? Somewhat like this type of structure, right? So how many proteins you will be able to see here? If you see this, how many you will find? Here you will be finding 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Now this one, I mean holding this completely, Stop. Sorry, I am not sure properly. Anyways, just remember, it's totally, it will be having 13 proteins like that. So they say that it is made up of 13 protofilaments. Filaments. What is this protofilament? Protofilament is nothing but one stack, one complete rack of this alpha and beta. It has formed a sheet. It has curled. It has, it has folded like this. But this one complete protein, if you see, how many are there like that? 13 are there. So this complete line, if you see, it is called one protofilament. So likewise, how many you will have? You will have 13 protofilaments. Are you able to see this? It's completely what? 13 I'm not shown 13 here. It has been, it is extra here. But just remember, it is made up of 13 protofilaments. So 13 protofilaments ultimately make what? Make this type of structure, and this is called microtubulin protein, microtubule, which is a cytoskeleton. Now this is width. I told about width, no, 25 nanometer, and that width is nothing but this only. Here, from here till here, if you see this, it just looks like a tube, no, like this. If you see this width total, it is 25 nanometer. That is why it is biggest or largest cytoskeleton. If you see, it is largest one. And what is speciality of this microtubule protein? What is the speciality of this is that, if this protein, if you just make like this also, if you see here, it is, it comes back, contracts, relaxes back. So this is the strength of this microfilament. This is the strength which makes it more stronger compared to the other ones. Okay. And That is why it will help in so many other structures. It helps in maintaining the shape, strength, motility. Mainly microtubules are the ones which gives motility for, for the cilia and flagella. That's what I told you. You need to remember cilia and flagella structure. It's completely made up of these microtubules only. So microtubules play very important role in structure of cilia and flagella. I hope you people are understanding this. So this is about protofilaments. So we have just completed one, that is microtubules. It is how it is made up of tubulin protein. Diameter is 25 nanometer. It is a sheet. It is made up of what? Alpha and beta tubulin, and then it forms a sheet, okay, and then it folds. After folding, if you see how many filaments are there, 13 protofilaments are there. They join together to form one microtubule. Clear? Next we'll be talking about microfilament and what? Intermediate filament. Okay, I hope you people understood this microfilament, right? Sorry, microtubule, right? So see, there should not be any confusion with this protofilament. Now when it forms when it folds, actually what you will find is what? You will find this alpha and then what? Beta, or you can say, like, I'll show it like this. Alpha, like this, and beta will be along with that alpha only, no? Always like this, beta. So like this, how many strands are formed? Likewise, you will find 13 protofilaments. Each one is called a protofilament. Above it will be blue then, like that. Okay. Such type, if you see, that sheet is made up of 13 protofilaments and that folds. And ultimately it gives a lot of strength. And mainly, as I told you, main function of this microtubules is in movement. It helps in motility. It helps in motility. In cilia and flagella structure, if you see, we are going to find this type of what? Microtubules only. So in that structure, we'll be talking in detail about this, how it is, that structure we need to study there. But I hope you understood this. If you understand this, then you will understand, you can imagine how cilia and flagella, their structure is made up of, all those things. I hope it is clear. Then we will do the next one. So it's made up of alpha and beta monomers. It is a dimer. Alpha and beta monomers, together it becomes what? Alpha and beta togetherly, we call it as a dimer. So many dimers are there ultimately. Okay. So this is about microtubules. Now coming to the next one, actually we need to study intermediate first, because intermediate is in between, no? In between. But anyways, it's our wish. So we will study microfilament. See, microfilament, you people, I hope you people are well versed with this microfilament. So this, what are the protein it is made up of? The protein it is made up of is actin protein, and some myosin also will be there. Okay, but mainly it is actin protein. And then, where have you heard this actin protein somewhere? If you have completed muscle chapter, what is that name of the chapter? I forgot. Muscles, when you have studied muscles, locomotion and movement, I guess. So in first PUC only, you have this locomotion and movement. In that chapter, you are very well versed with this actin protein. Anyways, we'll talk about that. Second thing, what you need to remember here is what? What is the diameter? It is about 7 to 8 nanometer. Fine. Now we will study the structure. You know it is made up of which protein? Actin protein. Now let us consider this is one actin protein. This is one actin protein. This is called actin protein, but this is called G-actin. You know why it is called G-actin? G because it is globular in structure, so we call it as G-actin. G-actin is a monomer. Okay. Likewise, so many G-actin will be joined to this, like this. So many G-actins will be joined like this. And then what? See, understanding this is very, very much important, because or else, okay, and this is G only, no? So you will see that it forms a structure like this. So many G-actins are joining together to form You are very much well versed about the structure in case of muscles chapter, wherein locomotion and movement. So can you see this? How it is formed? Like this. Again, it forms like this. So this is called now, this is a filament type structure. Yes, this is a filament like type structure. Now this completely, we call it as what? We call it as F-actin. It was G-actin, single ones, they are G-actin. Togetherly it forms a filament like structure and we call it as F-actin. Now one F-actin, it's like this, like DNA. It's like this. So this is beaded structure like this. This is one F-actin, one filament structure with all the G-actins like this. This is one F-actin. And what about this? This is another F-actin like this. I was not able to show it properly here, so I am showing this like this. This is one more F-actin. So two F-actins, no, curl together like this. How you will remember this? Just like you take some string with beads. You take two strings with beads and then if you just curl it like this, what will happen? Just like DNA, it will get ring like structure, no, like this. It forms a ring like structure and then this is how it looks. So these single monomers are called what? These single monomers are G-actin because it is globular in structure. Globular, that is the reason, G. And it forms a filament like structure or a fiber like structure. So we call it as F-actin. So two F-actin, when they join together, when they coil towards each other, then it forms protein. It forms a structure and that is called microfilament. Now what is the diameter of this? If you see this diameter or if you see this diameter, it is what? 7 to 8 nanometer. It is 7 to 8 nanometer approx. So this is about microfilament. And where you have studied this, I told you, in case of muscles, right? So this in muscles you have seen, also I told you, myosin proteins. So in this you will find that myosin will be, this is actin. I'll just rub this. So myosin will be present like this. Have you seen, have you observed it? Some heads will be there like this for myosin, like this. They will bind to this actin and pull it like this. In case of muscles, you have studied that, right? That is why it leads to contraction, relaxation. That is why we are able to contract and relax. The muscles help in contraction, relaxation because of actin and myosin only, ultimately, right? So this myosin, this actin protein along with some myosin, it forms microfilament. And compared to microtubules, width is little bit what? It is very less compared to intermediate also. So they are the smallest ones, you can say. They are the smallest, or you can say thinnest, cytoskeleton. Now what is their function? So to easily, as you know, in contraction and relaxation at least it will help. And the second thing is that in case of amoeba, if you have observed, you know, amoeba is very favorite organism for us. Why? Because it doesn't have any particular shape and size, no? Easily you can just go on drawing it however you want. These things are what? These things are called pseudopodia, right? False legs, pseudopodia. Their structure is not meant to be like that. But whenever they come across some things to move or to catch hold of some food or something, that forms, pseudopodia forms. Now there, this microtubules are there here. Wherever you will find that pseudopodia has formed because of this. Now what will happen is this myosin protein, wherever it moves, no, there that pseudopodia structure forms. Okay, so likewise what it will happen? It will help in formation of this pseudopodia in case of amoeba. So wherever this myosin, it goes on growing, no, if G-actins go on adding to this, it grows. If it is removed, it becomes small. That much only. So this is about microfilament. So it is again, function is what? Mechanical strength, support, maintenance of the shape. Also it helps in these things. Some kind of motility, you can say, some kind of movement. Okay. So this is about microfilament. Coming to the last one, this is also important, okay, for your understanding. If you understand microtubules and microfilament, that's more than enough. But still, we have one more. Now let us finish it off. So the third one what we have is what? Is the last one, intermediate filament. Intermediate filament. Okay, now I'm really literally sorry for the disturbance. Okay, and we are staying in such a place wherein we cannot tell anything to anybody here, so please cooperate until we are with this online classes. So we need to cooperate with this. Anyways, so intermediate filament, if you see, they're intermediate in case of size also. Okay, what is their size? And what is the protein which is present here is family of keratin protein. This is one thing. Second thing, if you see size, if you compare, size is what? I told you 10 to 12 nanometer, right? So around 10, you can remember 10 nanometer. Okay, then intermediate filament, what is the function where it is found? The best thing to remember this intermediate filament, we are not doing structure as such, it's not that important. So anyways, it looks like a fiber, and the main function, if you see, this found in the nucleus. Okay, this is nucleus. You know, nucleus is made up of this type of two membranes like this. I'm just showing you the rough one like this. Now we have done nuclear structure properly, but still inner to this nuclear inner membrane we have this fibrous structure like this. We have not done this. We have not done this there. So this fiber like structure, what you find densely, it is packed here. This is called intermediate filament. You know, this is intermediate. It is made up of intermediate filament, and particularly what we call it as nuclear lamina. What we call it as nuclear lamina. This is the third layer. I mean, after the first outer layer, inner layer, the innermost one is nuclear lamina. Completely fibrous it is. It is made up of this intermediate filament. Now this, which intermediate filament, which protein of this keratin? You know, it is keratin. So many types of keratin are there. Here, nuclear lamina is made up of lamin protein. Which is the protein here? It is lamin protein. So this nuclear lamina is made up of lamin protein. Lamin protein is a type of keratin protein. And finally, what is the function you can see here? Because it has this nuclear lamina, which is made up of this lamin intermediate filament, it has that particular shape. Nucleus have that, and this is nucleus, right? Nucleus have this particular shape because of intermediate filament which is present within nucleus as a third layer. Nuclear lamina we call it as. Understood? That is this intermediate filament. You know, if you compare those two, it is not dynamic in function. It is very much, you can say, very much strong, or what we can tell is that yeah, for example, if it is present here, it will not allow more, it is not flexible, we can say. It is not flexible. It keeps this nucleus like this only because of this filament. It remains like this only. Movement as such, it is not much important in movement or something. It is very much stiff, you can say. Good word is stiffness. It is stiff. It's not flexible like the other ones, other two. So this much about intermediate filament. So I hope cytoskeleton types we have done. Please don't think that NCERT has given only 3 or 4 lines and she is talking about more than 1 hour about these things, or more than 40 minutes about these things. So actually these things are important, because if you don't understand these things, then the next things what we study, that you will not understand. So basic things are more important, and in NCERT those people, basic things they'll not give. All the ones which is more important will be given. That doesn't mean we will just explain you that. If I just jump there directly and explain the structure of cilia and flagella out there, you people will not understand that, because you don't know about microtubule. What is microtubule? You don't know what is microfilament, how these are different. If you check NCERT, only 3 or 4 lines is given of cytoskeleton. Yeah, that you will understand it easily. That's okay, but structure you need to understand. If you understand here, then it becomes easy for you for the next class. Okay, we will see next about structure of cilia and flagella. So before coming to that class, you need to be perfect with, at least you need to be much perfect with which one? Microtubules. Okay.